I think we'll get started, so thank you for coming. My name's Anthony Wood. I'm a cell signaling technology, and I'm here with Ernest Heimsath from Agilent. Today, we're going to be presenting a story about looking at SMAD signaling using antibodies from cell signaling technology and the Citation C10 confocal imaging device from Agilent. So a little bit about us. I'll start off. I'm with Cell Signaling Technology. We're an antibody company primarily, and we are based in Danvers, Massachusetts, and it's our mission really to provide reagents for the research community so that they can advance the research, accelerate biological understanding, and ultimately deliver cures for human diseases. We are very proud to be recognized last year by the CIDAB organization as the recombinant antibody supplier of the year, and also the supplier succeeding in cancer research. So we're widely recognized for the quality of our antibodies, and that's will be the foundation of my part of today's presentation. Now I'll turn it over briefly to Ernest here to introduce himself.
Hello, so my name is Ernest Heimsath, and I'm representing Agilent, which is a global leader in the life sciences diagnostics analytical laboratory technologies. We are expanding very rapidly in the cell analysis space. I'm actually we were part of a Biotech Instruments before this, so that's where I'm coming from. So our fleet of imagers and plate readers, plate washers is really what we're bringing to the table and strengthening Agilent. So we're headquartered in Santa Clara, California, although I'm based in Burlington, the greater Burlington area in Vermont. As that's where Biotech Instruments is based, so we manufacture all of our own instruments. I'm in the applications lab, so it is my duty to demonstrate the utility of all of our instruments and provide support for our field team with any kind of scientific or technical challenges they may be facing. Thank you.
All right. So, and representing CST, if any of you were at the keynote address this morning, there was a discussion about the prevalence of poor quality data that is informing a lot of the models and AI's attempts to sort of overcome that. Well, from CST's perspective, we never want to be a contributor to that poor quality data. It's been our mission since the beginning to provide reagents that allow researchers to generate high quality data and with no concerns about its validity. And so we've been applying validation principles to our validation of antibodies for years, long before it was recognized that there was a reproducibility crisis in research. And we apply a number of these principles that we summarized in our hallmarks of antibody validation using a number of different principles, all applied in the application context in which the antibody is validated, that serve to demonstrate the antibody's specificity for its target. So these include right from a rational antigen design strategy at the beginning through to a variety of different testing models to ensure that the antibody is detecting its target and only its target. And again, it's important, and something that I'll come back to a little later, is these principles are applied independently in each application for which the antibody is validated. So today, the focus of the talk from a biological perspective is a TGF-beta signaling pathway. This really represents a constellation of many different signaling pathways represented in this diagram to the left here. There are over 30 ligands that can be, or ligands that activate multiple pathways that really constitute this constellation. It plays very highly complex roles in both development and disease, right from embryonic patterning through to developmental or late-stage diseases such as oncogenesis. It's also known to be a tumor suppressor. It contributes to the progression of fibrotic diseases, and it's also a significant regulator of the immune system, which is something that is now bringing more and more attention onto this pathway as we look for ways to manipulate the immune system to treat disease. So its clinical and translational relevance is quite high. There are currently 59 active clinical trials involving TGF-beta. This is from clinicaltrials.gov, and there's another 188 clinical trials that are recruiting currently that include TGF-beta as part of the trial. And these include small molecules, vaccines, monoclonal antibodies, etc., various modalities involving TGF-beta signaling at some level. So just an example, in the context of cancer, there's a process called epithelial mesenchymal transition. It's a developmental process, but it's very similar to what happens during cancer metastasis when epithelial cells lose their epithelial morphology and their phenotype and become more mesenchymal in nature, allowing them to become migratory and then, of course, undergo metastasis. And TGF-beta, as indicated over on the left here, is a major driver of that pathway that actually leads to transcriptional changes that allow that epithelial mesenchymal transition. This has actually been known about for a fairly long time. One particular molecule called HMGA2, a transcriptional modulator, is under the control of TGF-beta. It uses HMGA2 to elicit that effect through effects on transcriptional regulation. We can actually show this. This actually came from an antibody discovery campaign showing how when you treat cells with TGF-beta, it actually elicits a nuclear translocation of the HMGA2 protein, allowing that those transcriptional changes that drive the EMT. Also, in the context of immune system regulation, as I talked about earlier, there are various aspects within the tumor microenvironment in which TGF-beta signaling can have an input. This includes various different types of immune cells found in the microenvironment, all of which are under the influence of TGF-beta, often in an immune inhibitory way. In addition to that, there are other aspects of the tumor microenvironment, such as the angiogenic response elicited by the hypoxic environment of a tumor, that are responsive to TGF-beta signaling. In particular, the matrix metalloproteases that are responsible for remodeling the extracellular matrix during angiogenic growth are under the influence of growth factors such as TGF-beta and BMP. Now, as I mentioned, there are multiple pathways associated with TGF-beta signaling. You really have two primary axes, which are those driven by TGF-beta family ligands and those driven by the BMP family ligands. They have their own unique set of receptors, two for each of them. And then in addition to the canonical pathways which converge to the SMAD proteins, there's also an additional set of pathways, what we call non-canonical signaling pathways, such as ERK-MAP kinase, PA3 kinase, AKT, etc. These all cooperate with the canonical SMAD pathway to elicit the transcriptional response. So they're responsible for fine-tuning or modulating this pathway, which is really primarily driven by the SMAD proteins, which I'll talk about next. So the canonical pathway involves these SMAD transcriptional activators. And these are proteins that, upon ligand activation of the receptor, result in phosphorylation. Phosphorylation, first of all, of the receptors, but then that triggers phosphorylation of the SMAD proteins. If you're stimulating with TGF-beta, you're phosphorylating SMAD2 or 3. If you're stimulating with the BMP ligands, then you're phosphorylating SMAD1, 5, or 9. These phosphorylated SMAD proteins then recruit a co-SMAD, SMAD4, and this complex of either the SMAD2, 3, and 4, or the SMAD1, 5, and 9, and 4, then translocates down to the nucleus where it activates transcription of target genes. So if you are looking for readouts of TGF-beta pathway activation or the SMAD pathway activation, really what you are looking for is SMAD phosphorylation and SMAD translocation to the nucleus. It's often interesting. We get a lot of questions about ways of looking at activation of TGF-beta, the ligands, but really what people should be looking for is the readouts, which are the intracellular phosphorylation and translocation readouts that represent pathway activation most effectively. So if you're doing a molecule screening campaign, for example, these would be the same readouts you would likely be looking for as you're looking for phosphorylation or translocation of the SMAD proteins. So what do you need to do this? In order to look at phosphorylation and translocation, you obviously need target-specific antibodies. That's where cell signaling comes in. But no more than that, you would need phospho-specific antibodies. And that is our specialty and has been since our founding. And then you need them validated in the unique application in which your assay is relevant. It may be a Western blot, but more likely in the high-content scenario, you're looking at something like an ELISA assay or a high-content analysis platform using IF-validated antibodies. Again, I'll come back to this point. If you have antibodies that are validated in one application, it means nothing about whether or not they are functional in another application. So we go to great pains to independently validate our antibodies, and we only approve them for applications in which they have been validated. So this is just a subset of some of the antibodies that we have. These are primary antibodies that you could use for various applications. You'll see at the top there's a SMAD4 antibody. That's the co-SMAD. You can see from the dark, what we call chiclets here, this is validated independently in 6 different applications. And that's a rare case when you have one antibody that does work across all applications. As you can see, most of them work in distinct applications. They've likely been tested in all of them, but we only approve them for those in which they work. So you have to select the appropriate antibody for the assay context in which you are looking. Similarly, we have a number of ELISA assays to interrogate the TGF-beta signaling pathway. We have 2 different types of ELISA kits. We have PathScan ELISAs and we have FastScan ELISAs. And again, so if you're looking for an ELISA assay against that, we do have a number of options available to look at either the total or phosphorylated SMAD proteins in lysates. And I ranked that last list according to the number of citations. You can do that on our website by looking at our products by their citation number. We work with CiteAb, so that they curate citations in the literature for reagents, and you can see here that we have an abundant number of citations. Just searching for SMAD2, over 740 when I did this search about a week ago. But of course, that isn't in any context. Is that a lot or not? It really depends when you look at the whole constellation, which we have that data at CST. And if you do literature citations for SMAD2, you can see that cell signaling technology is dominant in that regard by a wide margin. The rest of these are other suppliers of SMAD2 antibodies. So we really have established a really strong reputation in this space based upon our expertise in signaling antibodies. When it comes to ELISA kits, again, I mentioned we have 2 different flavors of ELISA kits available. We have PathScan, a traditional sandwich ELISA kit that uses CST antibodies. And then we also have a faster, more rapid protocol called the FastScan ELISA. And again, similar to application validation, an antibody or a pair of antibodies that are validated in one application may not be actually work in another application. And this applies to the ELISA formats as well. So we independently validate for those 2 different types of ELISA kits. And this is just some example of some ELISA data and Western data at the bottom confirming the ELISA results that we get from the kit. So we have a whole resource page on our website if you're interested. A lot of people don't realize that we do have quite an expansive ELISA portfolio, so you can actually visit our page and look at, we explain all the differences in our protocols and various troubleshooting tips, etc. as well. Now, if you're looking at in cellular assays, you want to look at either biochemical or cellular assays using primary antibodies. Then again, we have a variety of antibodies. We have phospho-SMAD2 specific antibodies, phospho-SMAD3 specific antibodies, and we even have antibodies that detect both of those proteins due to the similarity between the sites. And then the clone ID is indicated in the parentheses, so you can see different applications for different clones are represented on this slide here. This is CHIP data. This is chromatin immunoprecipitation data for one of our antibodies demonstrating its functionality in CHIP. But again, not all of our clones would work in every application. Here's some examples. If you were stimulating with the BMP ligands, then you'll be looking at SMAD1, 5, or 9, phosphorylation of those SMADs. And so here's some examples of antibodies that work in IF, in Western blot and IF assays, 2 different clones that work in IF. And what you can see is upon treatment with BMP2, then you have an increase in the nuclear signal of the phosphorylated SMAD proteins. And that is representative of activation of that pathway through the BMP treatment. And then again, I mentioned SMAD4 being validated for multiple applications. This antibody is unique among the portfolio because it's the one SMAD antibody that we have been able to develop that works in a tissue or 3-dimensional context. And so it works in all 6 applications for which it's validated, but in particular and important and germane to today's presentation is it doesn't work in a 3-dimensional context and tissue context, and it seems to be predictive of its use in a spheroid model. So using these CST antibodies, we have partnered with Agilent, a really nice partnership. Combining our state-of-the-art antibodies with our state-of-the-art instrumentation, and our purpose was to demonstrate the quantitative evaluation of responses to TGF-beta pathway activation at different biological scales. So from the biochemical, looking at a SMAD phosphorylation through ELISA, in lysates, but also at a cellular level, looking at SMAD phosphorylation and translocation using fluorescence wide-field microscopy in cells, and then lastly, using a three-dimensional model of angiogenesis using fluorescence confocal microscopy. This, again, all done with the Citation C10 instrument from Agilent. So now I'll turn it over to Ernest here, and he'll describe the details of that.
Thank you, Tony. So I'm going to go ahead and use this slide also, and just to outline there, what I'm going to be discussing is, as Tony just described, is applications that use all three of these different levels of biology. So we'll start with the biochemical, then go through cellular, and then finally we'll end at the applications using a 3D model in HEVEX. But first, what I wanted to do is just introduce the system that we're going to be using to do these assays in, which is our Citation, our Agilent Biotech Citation C10 confocal imaging reader. So this is a very comprehensive and versatile microscope, but it's not just a microscope; it's also a plate reader that uses quad monochromators. And today what we're going to be showing is an ELISA that just simply uses absorbance, but it has much more capabilities beyond that as well. It also has three different modes of imaging capabilities. You can do transmitted wide field light, wide field fluorescence, as well as confocal fluorescence as well. So coupled with this is our software, Gen 5, and what this really does is not only does the C10 automate the imaging, but combined with Gen 5, which really automates the analysis. So you can really take your data, do the analysis, and get ready to publish graphs or images. This data can also be exported if you so choose, and but standalone, it has full functionality to calculate any of these metrics that you would be interested in. We're actually going to focus on this one kind of buried in the middle, this EC50 right here, and generate dose response curves that are going to look like this. This is also a 21 CFR Part 11 compliant. So if you're in a GXP laboratory setting, we provide that level of security and accountability in terms of the auditing capabilities and tracking that is required in that space. Okay, so the first part is the biochemical response, and in this way, as Tony had described, we're going to measure the SMAD2/3 phosphorylation, which is triggered by TGF-beta treatment. And I'm going to use two different kits from cell signaling. One is the PathScan ELISA kit that measures phosphorylated SMAD2 and SMAD3. I'm also going to use the PathScan total SMAD2/3 to quantify total SMAD protein. I'm going to use the Citation C10 in plate reader mode at absorbance 450, since these kits are based off of HRP conjugated secondary, which will give you an absorbance value. So first, just to review what we're looking at here, upon binding of TGF-beta to it, and of course, instead of Tony's very elaborate model, I'm just going to simplify this down for what the readout is. And upon TGF-beta binding to its receptor, SMAD2 gets phosphorylated. So the way we set up this assay is we would seed cells in a microplate, either of your choosing. In this one, I set it up in a 96-well format. Cells would be serum starved for a period of time. I do it overnight, about 18 hours. At which point we're going to treat them with a growth factor, TGF-beta, harvest those cell lysates. Then we're going to add those cell lysates to our PathScan ELISA kit wells, either the probing for the phospho-SMAD2 or total SMAD, develop our HRP signal, and then stop the reaction. Well, we're going to get a colorimetric absorbance readout. We're going to measure that, absorbance 450 nanometers, and then generate our dose response curve. Now the software, once you get your raw data, this is what it will look like in our Gen 5 software. So the power of Gen 5 is that you can take this raw data and you have a list of different calculations at your disposal that you could choose to transform that data, either from the raw data blanket, if you have designated blanks. You can convert it into some other format. For example, for these ones, I report not just the raw data, but like a fold change compared to control. And so this is what it looks like. So if we do a dose response curve across these multiple logs, across a multiple log scale of TGF beta, what we see is that we get a nice dose response of phosphorylated SMAD2/3. From this, we could generate our EC50, which is 34 picograms per mil. But importantly, since we use this in combination with a total SMAD, we could compare this data with what our total, essentially total SMAD2 load is. And if you're more familiar with Western blots, this could be like your protein load. That what we can do is then adjust these values based on wherever the protein is, which is actually what these numbers are representing, is an adjusted value based on the protein concentration that we quantified based on the total SMAD ELISA kit. So while an ELISA is powerful because it's very sensitive, what it cannot do is provide any kind of spatial information, such as we know that a phosphorylation event happens. We don't know where that happens. But using an imaging technique can provide that insight. And so what we're going to do next is transition to the cellular level of analysis, is going from cultured cells, where we're going to use a phosphoantibody to image where phospho-SMAD is located. We're going to use the wide field fluorescence modality of our Citation C10 and quantify that change. So here we are again, and these are the reagents that we're going to use. So we're going to use cell signaling phospho-SMAD2 antibody, as well as for TGF beta signaling, but also for BMP signaling. We're going to probe for SMAD1, an antibody that recognizes SMAD1, 5, and 9. That's phosphorylated at these serine residues. We're going to do this, and our cell model is going to be these immortalized human fibroblasts that we source from in ScreenX in Germany. These are great because these are a nice non-disease-derived cell line. So if you want to get what's maybe a more reflective responses and not a disease state, I really like using the cell line. And again, we're going to use our C10, this time in a wide field fluorescence mode. So at a simple kind of a plus-minus experiment, after we serum starve these cells cultured in a 96-well plate, we see when we probe for SMAD159 in a control, we see no nuclear signal. However, that signal was dramatically increased over an hour of treatment at 100 nanograms per mil of, in this case, BMP2. Now, what we can do is, if we also the red is a counterstain with microtubules, just to kind of show cellular context of where cells are. Also, as a counterstain, if we use DRAC6 to visualize the nucleus, using the Gen 5 software, we can identify these nuclei, create a mask, and then quantify GFP signal within that nuclear mask that we have created. Doing so, we specifically focus on what is the nuclear signal for phospho-SMAD159 when it is treated with BMP2. So this is a nice demonstration of a plus-minus drug treatment. Now, what we can then do is use the automated features of the Citation C10 to expand this out into a much broader array. If you want to get a little bit more in-depth insight into the potency of, say, BMP2 or TGF beta, which we would want to do is do multiple concentrations. But to be able to do this on multiple samples and larger than just 2 samples, we need a way to be able to reliably image them correctly. Oops, sorry, Ryan. Oh, sorry. Let me just say this is also repeated with TGF beta. So I showed you data for BMP2 with TGF beta, the same thing, 0 and 100 nanograms per mil. We get a response. We can quantify that response as well. So if you were going to translate this and scale this up to a high throughput format, instead of just 2 sample comparison, what we will need is a way to image reliably one well from the next. And that's where we have what's called laser autofocus, laser-based autofocus. And essentially what we do is we shine a laser at a particular angle that triangulates that Z height in any given well, which may vary from one well to the next. But in doing so, we're able to reliably image an entire 96 well plate, and then we could expand that from 1 or 2 samples to multiple samples. In this case, I set up a dose response with the highest concentrations here down to our lowest concentrations in our citation C10. And with that, we can now take not only a 0, a control, and a given concentration, we can then titrate in either BMP2 or TGF beta and get a nice dose response and derive our EC50s based on this. So now we have 2 methods. We have our ELISA, which we can show a phosphorylation event happens, and now we have spatial information that show not only did it happen, but it happens, and we can quantify that species, that phosphorus species, in the nuclei of these treated cells. Now, if we expand this even further to our last scale, or at our highest scale, which is essentially a tissue level or a 3D level scale, what we're going to use now is our confocal capabilities of the C10. And this is an angiogenesis sprouting assay using HUVEC cells. I'm going to use that same antibody that Tony had mentioned, the SMAD4, that's validated all the way from a biochemical assay and an ELISA to cellular, and now that has been validated in a tissue level or histological level analyses. The cells that we're going to use, instead of those fibroblasts, we're going to use HUVEC cells that were sourced from Lonza. These are pooled donor HUVEC line that they supply. And again, we're going to use the citation C10 in confocal mode to evaluate SMAD expression in these. And if I'm not sure if you're familiar with these angiogenesis models, but what you do is you form a spheroid of HUVEC cells, and then you transfer that and you suspend them in a fibrinogen matrix. So these are suspended in a fibrinogen matrix, and in this case, what I did these in a 96 well as well. Once that matrix is formed and solidified, you overlay a feeder layer of fibroblast cells that provide additional nutrients beyond what is in the typical growth medium. And after culturing for a few days, this was at day 3, those HUVEC cells will start to sprout and start to form what is kind of like an in vitro replica of blood vessels, essentially. And so this is a fairly popular established model for understanding mechanisms of angiogenesis. And again, we're going to image this in our confocal mode of our citation C10. And I just want to say a few things about confocal fluorescence. And essentially what it does is it really cuts out on background out of focus light, which is really crucial when you're looking at thick biological samples. And it does so, sorry, this went a little bit. It does so by, so this animation I'm not going to worry about, but by passing a laser light through this array of pinholes here. The excitation light will eventually hit and excite the whatever fluorescent probe in your sample. That excitation light will return back and pass through those same pinholes and go to your detector. In this case, we're using a Hamamatsu camera here. Now, the purpose of these pinholes is to, that laser light is still going to excite all fluorophores on multiple Z planes. However, that pinhole, what it will do is it will essentially cut out any kind of autofocus light from every other Z plane except where your Z focal height is, as demonstrated here. And these are 4-micron beads. And what I hope you can appreciate is in wide field, you get all this autofocus light that looks like columns. However, these beads show up actually as spherical beads in confocal mode. And so this is really a crucial concept in being able to image 3D tissue, or in this case, spheroids. As an example, here is, this is a movie that's going to play, and it's in 3 colors. The green is going to be our SMAD4 antibody stained, and then also a counter stain, which is a endothelial-specific marker, PCAM1, also called CD31. And then the nuclei are in gray. But as this movie plays, what it is, it starts at the most bottom level of that spheroid and pans up in the Z plane and shows the 3-dimensional nature of this sample, of this spheroid. And here, what I did is I just flattened all of those those Z slices into one image, a maximum intensity projection. As this was made up of 187 different slices that make up this spheroid. That thickness is also just under 200 microns thick. So what I hope you can appreciate is, even though we're imaging this thick sample, it's what confocal mode can do is just really make this a very clean image in terms of removing a lot of noise that would fluorescent noise that would be in your sample. Now, what you can do is take an image like this and really drill in to look at localization events that are occurring. So if we focus in on this one region of the spheroid, we see these different angiogenic sprouts made up of these endothelial cells. And at the tip, you have multiple cells, but at the tip, they're called tip endothelial cells. And they kind of really lead the way and are responding to growth factors that are in the media and essentially grow towards them. They chemotax towards them. Proliferation happens in the stalk cells, which make up the rest of the angiogenic sprouts. So if we look at these different channels, what we can see is these sprouts are indeed expressing SMAD4 as well as PCAM1, and then we can do, so now what we can do is start thinking about different ways to probe and to understand this system and the mechanisms of angiogenesis by starting to ask questions like, what happens if we stimulate with TGF-beta or BMP6 to our SMAD4, and also counterstain. And now that we have a nuclear signal, we can start to look at some of the other downstream signaling events from TGF-beta or BMP signaling. But we'll leave it at this right now. And so what I want to do is just bring back and summarize everything I've shown you, which is first, we talked about how we can use the Cytation C10 with cell signalings, the PathScan, as well as the FastScan. I showed the PathScan ELISA kits, but certainly the FastScan work as well on doing ELISA and getting biochemical insight into a cellular response, such as phosphorylated SMAD. We can then, at the next level up, is look at a cellular event and get spatial information, such as how and when those phospho-SMAD species exist and where. And lastly, we can look at a 3D model and demonstrate that those SMADs are co-expressed in our 3D model as well. So with that, I'd be happy to take any questions. We have a few more slides, and please visit us at our respective booths. Cell signaling is at 2530. We're at 1811. But also stay connected with us too. We're on different forms of social media, as well as we have different resources available if you want to learn more about both our system, as well as any new applications that we generate along the way. So thank you, and we have a similar slide for cell signaling, which Tony will take.
Yep. So similar format. So just if you have any questions about our portfolio of antibodies, please don't hesitate to drop by our booth. Happy to answer any questions. Of course, we have much more than just TGF-beta portfolios. Got over 4,000 antibodies in our portfolio, and many of them focused on the signaling space. So drop by. Happy to answer any questions. Thank you.